All right, welcome back to The Explainer. Today, we're diving right into the microscopic heart of our cells to look at one of the most amazing processes in all of life, how we copy our DNA. And we're not starting in the middle. Oh no. We're going straight to the very, very beginning, to the starting gate.
So I want you to really think about this for a sec. Your DNA is basically a 3 billion letter instruction manual. 3 billion? How on earth does your body make a perfect, flawless copy of that entire thing over and over without messing it all up? I mean, it is a truly mind-blowing feat of engineering.
Well, the answer to that huge question actually starts with something incredibly simple. Before you can copy anything, a book, a hard drive, whatever, you've got to know where to start. And for DNA, finding that one single correct starting line is absolutely everything. That's what we're talking about today. Okay, so where is this super important starting point? What are we even looking for on that long strand of DNA? It's a special spot called the origin of replication, or just the ORI site for short. And an easy way to remember that is just to think ORI as in the original point. This is it. This is the official, non-negotiable starting line where this whole race is going to kick off. But just finding the starting line isn't quite enough. DNA is a tightly wound double helix, right? You can't just start copying it. You need a specialized team to get in there and prep the track. So let's meet them. We're going to call them the unwinding crew. First up for the crew, we've got the one that gets this entire party started.
These are the initiator proteins. In simple life forms like bacteria, it's called DNA. In more complex organisms like us, it's a whole group of proteins called the ORC. But their job is the same. They scan the DNA, find that special ORI site, and clamp on, basically planting a giant flag that says, "Start copying right here." It's the DNA awakener, because it literally wakes up the DNA and tells it it's time to replicate.
So the spot is marked. Now the crew needs to actually get inside that double helix. And for that job, they call in the muscle of the team. This is an enzyme called helicase, and its one and only job is to just power down the DNA and unzip the double helix, breaking all those tiny little hydrogen bonds that hold the two strands together. The name is perfect, right? Think of it as helicase. It literally opens the DNA case. Now, once the DNA is unzipped, a new problem pops up. Those two single strands are, well, they're kind of sticky. They desperately want to snap right back together, so the crew needs to bring in some safety equipment, and fast.
And that's where these guys come in. Single-stranded binding proteins, or SSBs. They just swarm all over the newly separated strands and hold them apart. They act like little safety seatbelts, keeping the strands secure and stable, so the copying machines can get in there and do their thing.
Okay, quick recap. The starting spot's been marked, the DNA case has been unzipped, and the strands are being safely held apart. The stage is officially set for the main event. All this activity creates this opened-up section of DNA that looks, well, it looks just like a bubble. So, of course, scientists call it the replication bubble. And at each end of that bubble, you've got this little Y-shaped junction where helicase is still actively unzipping. That spot is known as the replication fork.
We are almost ready to go. There is just one last piece of the puzzle. The main enzyme that actually builds the new DNA is a bit of a diva. It's a master builder, but it can't just start on a blank slate. It needs a clear signal telling it exactly where to begin. So the cell calls in the boss. And that boss is primase. Its critical job is to lay down a short little strip of RNA, and we call that a primer. This primer basically acts as a giant flashing go sign. It provides the perfect docking station for the main DNA copying enzyme to land and start working. That's why we call it the prime minister. It gives the first official order to start building. So if you put it all together, this whole setup happens in this beautiful 4-step sequence. First, you've got origin recognition, then DNA unwinding thanks to helicase. After that, strand stabilizing by the SSBs, and finally, RNA priming to kick things off. That perfectly choreographed sequence, that's the whole process of initiation. Now, this process is fundamental to all life on Earth, but nature has a couple of different strategies to get it done. So let's do a quick comparison between simple organisms, like bacteria, and more complex ones, like us. If you look at a simple bacterium like E. coli, its DNA is a relatively small circle. So to copy the entire thing, it only needs one starting point, just one ORI site. That's it.
But what about us humans, with our huge linear chromosomes? Well, we have thousands of origin sites. The difference in scale is just staggering. We have to do this, right? To copy all of our DNA quickly, we have to start in thousands of places at once. It's like having thousands of construction crews working on a giant skyscraper all at the same time. So here's the breakdown. Prokaryotes keep it lean and mean. A single ORI site, the DNA protein, and the whole process is incredibly fast. Eukaryotes, on the other hand, are more complex. We've got multiple ORI sites, the more complicated ORI-RC proteins, and the entire process is way more regulated and intricate. Just two different strategies to solve the same problem.
Okay, so we've gotten into the how, and it's fascinating. But let's zoom out and talk about the why. Why is getting this one single step, the start, so unbelievably important for a cell? Because everything that comes next depends on it. A perfect start makes sure replication begins only at the right place, the ORI site. And just as important, it only happens at the right time in the cell's life cycle. If you mess this up, if you start in the wrong spot or at the wrong time, you risk copying the DNA wrong, maybe not finishing it, maybe copying it too many times, and that is a direct path to harmful mutations. So yeah, a perfect start is non-negotiable for survival. All right, I know that was a lot of molecular details and names. So how can you possibly remember the key players and what they do? Well, we've got a really simple memory trick that captures the whole sequence perfectly. Just remember this sentence. Old Harry said, please. The O is for origin recognition. H is for helicase unwinding everything. S is for the SSB proteins that stabilize the strands. And P is for primase, which adds the primer. Old Harry said, please. Simple as that. So, one last time, let's boil it down to its absolute essence. The origin is recognized, helicase unwinds, the SSBs hold, and primase primes. That's the core sequence. That's life's starting gate. So, just let that sink in for a minute. This entire incredible molecular dance we just walked through, it is happening inside your body trillions of times over right now as you listen to this. And it's just the very first step. That's the really crazy part, which kind of makes you wonder, doesn't it? If this unbelievable machine